Welcome. We're to the point in our study of our microbiology text by Tortora that we can take a look at an organ system and the diseases associated with it. We've learned a lot about history of microbiology and a lot about the technical aspects of microbiology and various general mechanisms of disease and so forth, and some of the microorganisms in different categories and classifications that cause disease. Now we're going to look at the skin and eyes and look at some diseases, and all of the things that we'll look at today, as far as I'm pretty sure, will be a review of things we've mentioned before, just to put them in this context. So there's our micrograph of great beauty for the opening of this chapter, some Pseudomonas aeruginosa, which we'll mention briefly in this chapter. This is pretty much of a quick survey. We won't add too much detail to what we already know about these microorganisms. A little bit about the skin again. Perspiration and sebum provide nutrition on the one hand, so they support microbial growth, but yet lysozyme is found in sweat, which inhibits microbial growth. It hydrolyzes, breaks down the peptidoglycan cell wall, and so that's kind of deleterious to microorganisms, keeps them at bay. Fatty acids also have an acidic pH, and the sebum, I should say, has an acidic pH, and some of the fatty acids are not very nutritious for some microorganisms. So there's a few microbes that can grow on the skin or live on the skin, but they're pretty limited in what they can do and in the range of species. Antimicrobial peptides, also defensins, are produced by the keratinocytes that make up the outer boundary of the skin, the epidermis. So that's another thing that kind of limits bacterial presence on the skin and certainly helps keep them from getting through. Very few microbes can penetrate through the uninjured skin. Here's a little cartoon schematic diagram of the skin. There are bacteria that colonize outer parts of the hair follicle and the sweat gland and the sebaceous gland. Here's the sebaceous gland. This is a little oil gland that's making the sebum, that oily stuff that we said is kind of acidic that comes out onto the surface right there. So those are routes that some microorganisms can't get into the skin, especially in tropical climates where the skin is always moist and damp and it's very, very hot. And there's some pretty aggressive critters down there as well, microbes. Just briefly, this is not really the subject of our chapter, but mucous membranes are also epithelial boundaries between your body and the outside world, and their epithelial cells can also produce some antimicrobial peptides, defensins. The mucus that lines the surface of all of these mucous membranes, produced by either glands within the membrane or goblet cells, is somewhat antimicrobial. It tends to have immunoglobulin type A in it, those dimers of antibodies. It's acidic in pH. In the respiratory system, the cells, the epithelial cells, actually have cilia that can act like motorized whips and do the wave and sweep that mucus and whatever microbes that have gotten stuck into it right up out of the lungs or right posterior from the nasal cavity back into the pharynx, and it can be swallowed down into the stomach. So anyway, on to our subject of the skin. Moisture provided by perspiration encourages microbial growth. We just mentioned this is just for your own review. You can take a look and answer that question as you're going through the study. I got stuck there for a second. All right, so thinking of the microbes that live on the surface of the skin, gram-positive salt-tolerant bacteria, for the most part, Staphylococci, know that they're salt-tolerant. They can grow on mannitol salt plates. Micrococci, just another type of gram-positive coccus, and diphtheroids. They're a little bit, in some way, related to Crinibacterium diphtheriae, but they're just called diphtheroids. They're harmless bacteria that live on the surface. They can metabolize the oils and sebum, and so they have some source of nutrition there. Crinibacterium xerosis is a common member of the commensals on the surface of the skin. Propionibacterium acnes is an anaerobe, and that one ferments carbohydrates producing propionic acid, and those guys are associated with acne, mention them in a little bit. There are some fungi that can colonize the skin. Malassezia furfur may be found on the surface of the skin. May, for example, cause some dandruff, the flaking of the outer parts of the stratum corneum of the epidermis. Rashes are a very common feature of many diseases that either infect the skin or that have a presence on the skin when they become systemic in the body. Some viruses, when they become systemic and they're in the circulation, they get into the skin and cause rashes. So exanthems are rashes that are arising from a focus of infection other than the skin. And enanthems are mucous membrane rashes arising from another focus of infection. So we have skin rashes and mucous membrane rashes. Oftentimes, if you study a disease closely, childhood disease, for example, it'll mention when to peer into the oral cavity and look for a particular rash feature on the mucous membrane. So rashes come in different types. Some rashes are relatively just flat red patches. They're called macula. So a macular rash is just a flat rash with a different color than the surrounding skin, typically red or orange or pink. A rash with vesicles. Vesicles are fluid-filled little bulges, papules that stick up, and so a vesicular rash might have some fluid-filled risen parts. A pus-filled blister in the skin is called a bulla. Some vesicles are quite clear and they have no pathogens in there and no pus, and some do have pus in there, so that'll help us distinguish some diseases. A regular bulla, a pustule. I wasn't looking carefully where I was reading from. A pustule is a place where there's a lot of dead neutrophils and dead microorganisms and tissue debris forming a viscous yellowish fluid that's called pus, whereas a bulla is just a regular blister. We've already encountered bullous disease and talked about bulla like in the context of burns and so forth. So that's just a fluid-filled blister on the surface of the skin. Staphylococcal skin infections. Staph bacteria and strep bacteria cause the majority of skin infections by far. Staphylococcus epidermidis is coagulase negative and is found in large amounts on the skin. Staphylococcus aureus is a coagulase positive, gram-positive coccus that is found on an estimated 30% of people's skin, less common. So Staphylococcus aureus is the one that causes the diseases, and it can have a whole bunch of different virulence factors depending on what genes it has acquired, what factors or plasmids it may be harboring. And here's a list of some virulence factors that may be found in a particular strain of Staph aureus. One thing is it may be antibiotic resistant. We've talked about beta-lactamase or penicillinase that is very common now. Methicillin-resistant Staph aureus is a very common problem both in the clinical setting and the community now. Leukocidins are toxins that kill white blood cells, neutrophils. Some Staph aureus can evade phagocytosis. They have a protein on the surface, protein A, which binds immunoglobulins, and it binds them by the stem region so that they can't then bind to the Fc receptor on macrophages. We talked a lot about antibodies opsonizing microbes and then becoming activating phagocytosis. Well, this is a way that the Staph bacteria can prevent that from happening. They also have a capsule that resists phagocytosis, so these little guys are a little bit hard to chew up. They can also survive in the phagolysosome. They're lysozyme resistant. They're because of their capsule, so they're pretty durable. Toxins. Exfoliative toxin causes scalded skin syndrome. It causes the skin to actually split and peel, as we'll see in a picture. Leukocidins, we already mentioned up there, farther up. I think I got a little redundant here. Kills neutrophils. Toxic shock syndrome toxin. They're famous for producing a toxin that can influence your whole cardiovascular system, cause systemic inflammation and vasodilation and a big blood pressure drop, and shock is a condition, it's actually short for circulatory shock. It's a condition in which the blood pressure falls dangerously low and there's not enough blood delivery to the tissues, and it starts causing organ damage and organ failure. So toxic shock syndrome could be fatal. Super antigens, we talked about super antigens before. Some staff would use super antigens that just randomly activate helper T cells, and they become revved up and start producing a ton of cytokines. We get a cytokine storm, which is not particularly helpful. Some staph bacteria can secrete hyaluronidase, which is an enzyme that breaks down hyaluronic acid, a structural protein of the extracellular matrix of tissues, and start being able to penetrate into tissues and spread that way by breaking down the structure. And lipases allows them to metabolize lipids in hair follicles and glands and so forth, so that gives them more metabolic freedom and capability. And so antibiotic resistance is a big problem with Staph aureus, and you can see some different antibiotics on this plate. 4 antibiotics that don't do any good anymore. Methicillin, probably penicillin. There's another penicillin derivative, I think it's Xanicillin or something like that starts with X, probably erythromycin or something like that right here on the E. And then over here is vancomycin. Vancomycin is like considered kind of the last hope for some of these resistant bacteria, but the problem is now we're starting to encounter vancomycin resistant strains of different pathogens. Good thing we have a few more antibiotics that are newer and have slightly different mechanisms of action we learned about in the last chapter. So here's just a cool pair of micrographs that was produced as a biofilm, was produced progressively by some Staph aureus on a catheter. Here you can see the bacteria adhering to the surface of the catheter plastic, and some biofilm starting to form on the surface. And then here we've seen the entire thing is completely covered over by biofilm. Most of the bacteria that are present in this community are entirely ensconced, buried down in that layer of sticky polysaccharide there. So they're really, really tough to get at. They're very protected by that biofilm from antibiotics, from white blood cells, and so forth. So what kind of infections do they cause? Well, folliculitis. They can get down in the hair follicles and start causing inflammation down in there. And if you get a hair follicle infection, sometimes that's called a boil. If it happens on the eyelash, or on the eyelid, I should say, you get a folliculitis of an eyelash hair on your eyelid, that's called a stye. It'll swell up and be all red and so forth, and maybe even have some pus visible. A furuncle is an abscess, a larger pus-filled area, inflamed area that's gotten down into the hypodermis. So it's a follicle that got infected. That infection spread it down through the dermis, and now it's into the hypodermis, which is a layer of fat underneath the skin. So that's a growing problem here. A carbuncle is even worse. A carbuncle is when that happens and a number of follicles have been infected, and the infection is spread down into the subcutaneous tissue. And so now you have a pretty good size infection going on. It'll start to affect the body systemically. Fever and malaise, feeling of illness will accompany the carbuncle. Now these are things that, and the names are not familiar to us, and those are things that are probably memories from a time when before antibiotics were prevalent and weren't available to people who get an infection. It would just get worse and worse, and there wasn't really much they could do about it. Now you quickly treat things. You don't wait till they get to the level of a carbuncle. Impetigo is an infection. It's a non-specific title for an infection usually around the oral cavity of children. They have injuries, minor injuries to the skin, and they get some infection in there around their mouth, and they scratch. It gets itchy or chapped, and they scratch at the skin, and then bacteria get in there and continue to infect the skin. 80% of the time it's caused by Staph aureus, 20% of the time by Streptococcus pyogenes. Streptococcus pyogenes is worse because that same kind of superficial infection can worsen to spread into the tissue underneath the skin and cause what's called erysipelas in the case of strep. So that's pretty terrible. So let's take a look at it. Here's a little picture of some of these crusty looking lesions on someone with erysipelas or impetigo. I didn't even say erysipelas, impetigo. So we have this most likely staph infection of the skin where the skin's been kind of damaged, and then the bacteria have had a chance to get in there where the epidermis is not contiguous, and it starts to chew things up. Scalded skin syndrome is a more severe situation where the bacteria have gotten right into the skin, infecting the skin, and the toxin, the scalded skin exfoliative toxin is present, and it's causing the skin to peel right off because all the skin is infected now with bacteria. So it's a pretty serious condition for that poor little infant. Toxic shock syndrome, there's a toxin that produces that, and if enough staph bacteria are congregated in a particular place in the body where they produce a lot of toxic shock syndrome, that can again cause a systemic reaction, cause cardiovascular shock. And that's a very, very serious and potentially life-threatening condition. The first time I heard of it, it became famous probably around the time when tampons were invented. Tampons were a real solution for a lot of people who needed to be on the go, professional women who, when they had their period, they could just use a tampon to block things off for however long they needed to. And sometimes people left tampons in for a long time. They didn't realize there would be any downside to that. And if they got a staph infection in their staph culture going in that in the materials being shed from the uterus, blocked off by that tampon, then eventually a toxic amount of this toxin would be produced eventually, and some people died from it. And then after that there was a ton of warnings all the time about changing your tampons regularly and so forth. I know that's kind of a funky topic, but that is an important part of the history of understanding toxic shock and staph bacteria. There's a poor little guy. Streptococcus pyogenes. Here's just a quick thing about the classification of strep bacteria. There's a lot of different types of strains of Streptococcus, and sometimes it's hard to keep them straight. And so that's why I put this little chart in here, and you can refer back to that at a later time. You might even want to take this out and put it in a document or something if we're going through other chapters. But even I have a hard time remembering sometimes, wait, which is alpha hemolytic and beta hemolytic? Wait, which is a group A and group B? So anyway, alpha hemolysis refers to the tendency to partially lyse red blood cells that are, or utilize red blood cells growing in blood agar. So if you culture strep pneumoniae, or strep mutans, or strep mitis, those are what's called the Viridans group. That's a Lancefield classification or grouping of strep species. They will produce a greenish yellow area around the colonies amongst the red color of the blood agar. So they're utilizing that blood agar to some degree. That's called alpha hemolysis, and that's characteristic of these bacteria. Strep pyogenes and strep agalactiae represent the group A Streptococcus and group B Streptococcus of the beta hemolytic classification. Beta hemolysis means they completely lance the red blood cells. There'll be a clear area, a clear like brownish tan area of just the agar around the colonies in a blood agar plate. So strep pyogenes we've just been talking about in skin infections, and strep agalactiae is more of a problem in neonatal clinic. Causes pneumonia and meningitis in neonates. So that's a bad kind of situation, just so we can keep these things straight. So we're talking about strep pyogenes here for the most part. Group A strep, or GAS. Once you have learned that a lot, when you just see the word GAS, you'll instantly think of strep pyogenes, know exactly what we're talking about. All right, so what are some virulence factors that strep pyogenes may have that infects the skin? Hemolysins, enzymes that can break down red blood cells, gain them access to the nutrients as well as the iron. Hyaluronidase, we already counted that with staph, and some strep species carry genes for hyaluronidase that can break down the tissue structures. Some even have collagenases, and they can then spread right into tissues as they just dissolve some of the structural proteins of the tissue. Streptolysins are enzymes that can break down fibrin clots. I don't know if we mentioned, we should have mentioned, there's staph enzymes also that can break down clots. M proteins M proteins are found on a lot of strep pyogenes bacteria that can help them evade phagocytosis. They don't stick to the receptors on the phagocytes, and they can also help them survive with phagocytes. So that's an important virulence factor, super famous factor of disease-causing strep bacteria. Exotoxin A, it's a toxic shock syndrome like toxins, much like the toxic shock syndrome toxin from Staphylococcus aureus. And so it can also cause, if you get enough of these things congregated together, you can have a lethal intoxication with this particular poison. There's a micrograph showing M proteins on the fibrils of some beta hemolytic strep, meaning strep pyogenes, and some without. Here's a picture of erysipelas. So now we've got an infection, an infection in the skin. Now it's gotten into the tissue under the skin and is spreading along under the skin. So this well-defined red area, there's just quite a deep infection going on there. That's a very serious problem. It needs to be stopped quickly before it really starts to spread. And you'll see a horrible example of that in just a second. So there's a toxin that causes the redness there as the bacteria get deeper and deeper into this tissue. Necrotizing fasciitis. The fascia is the connective tissue that's between the fat under the skin and the muscle that lies beneath it, muscle and bone that lies beneath it. And in this case, strep pyogenes bacteria have gotten into that area where the fascia is. There's not much vasculature in there. They've gotten in there and started infecting and secreting their collagenases and their hyaluronidases and their streptokinase, and they're just spreading along underneath the skin, spreading along and just killing the tissue as they go. So what has to be done here is, on a periodic basis, the nurses and physicians have to come in here and debride this, remove all the dead tissue, because the dead tissue will cause microbes to grow and hinder healing. You got to keep on debriding all the dead tissue and try to keep it as sterile as possible and try to get some healing to start happening. There's no doubt that if this person survives, and they have about a 50-50 chance of that, they're going to need skin grafting to recover over that wound for sure. It's not a good situation. That's just, I'm not sure exactly that that's not part of the natural tissue. That's something that was inserted in there medically. Streptococcal toxic shock syndrome. Shock is a condition, a circulatory shock is a condition in which there's a huge drop in blood pressure. Again, as I mentioned before, the blood is not sufficiently delivered to the organs. They're hypoxic, they're ischemic, and so the cells start to die, and you're going to have multiple organ failure. So all kind of problems going on here. One of the reasons for the toxic shock syndrome is listed right here. We have massive activation of inflammation by these bacteria. We're going to turn on those neutrophils, and the neutrophils are going to start secreting cytokines, which are going to cause more and more inflammation. Inflammation, as you know, when you have histamine coming around, causes vasodilation. Histamine and kinins and prostaglandins and leukotrienes, they all cause vasodilation. Opening up the blood vessels, the blood pressure drops. Pseudomonads. Pseudomonas aeruginosa, we know about that one. We've mentioned before. It's this metabolic powerhouse of a bacteria, gram-negative aerobic rods. It produces a pyocyanine pigment. It's a blue-green color, so that when there's an infection with Pseudomonas aeruginosa, it tends to turn blue-green. That's the color of the pus, or of the surface of the tissue. So that's kind of interesting. I should have dug up a picture, and I didn't. Pseudomonas dermatitis, inflammation by Pseudomonas. You can have it in your skin. You can have it in your external ear, otitis externa, swimmer's ear. Right in the ear canal, you can have an infection with Pseudomonas. It's in the water. It's transmitted in water. That's not well enough chlorinated, and it's a common problem, causing a painful infection in the ear canal. It's an opportunistic infection that cannot pass through the epidermis. For all of its metabolic impressiveness, it can't pass through the epidermis unless there's an injury to the epidermis, and then it can get in there. Let's say, for example, a burn patient has lost a whole bunch of the surface area of their body, their protective barrier of the epidermis, and now they're open to Pseudomonas bugs getting in there, and you'll have a hard time beating those things back. They have some virulence factors that help them go to town once they get into the body, get through the epidermis of the skin. They have fimbria and adhesins, allow them to attach to the surface of the tissues. They can start producing a biofilm that will protect them from phagocytosis and antibiotics. They have a capsule to help evade phagocytosis. They have endotoxin or gram-negative, so they produce endotoxin, which is toxic to the host, that lipid A material. They produce exotoxins, A and S, which are toxic to host cells. Pyocyanin is that pigment that we mentioned up here that causes the blue-green color. It also causes cells to produce oxygen radicals, which can cause collateral damage to tissues. So, bad bugs, hard to kill. Acne. Acne is a disease that has various degrees of severity, and it's named differently for different classifications of acne. Comedonal acne is just when there's a few pimples here and there. Pimples are just hair follicles that are clogged because of the rapid turnover of keratinocytes, especially during adolescence. The androgens and estrogens tend to cause the increase in the rate of keratinocyte turnover in the skin, and they start to get the exfoliating cells get clogged into the follicles, causes what's called pimples, and then sometimes some bacteria get in there and grow in there in that clogged pore and cause inflammation. Now we have inflammatory acne. Some people have a very, very severe type of acne where there's very deep little mini abscesses in the skin going along. So mild comedonal acne, the sebum channels are blocked, but there's no infection there. White heads are sometimes called. Eventually the white colored sebum, the lipids, will oxidize in the presence of oxygen and turn black. Now you have blackheads, but they aren't infected yet. They're not all red around them and sore. Eventually, though, there may be some. But anyway, what we're going to do about those treatments, use retinoids to slow down the production of those skin cells, salicylic acid to help loosen the clogs in the pores and open them up so that the sebum can escape. If I look up adapalene, I can't remember what it is. You can check it and see what you see. Propionibacterium acnes is that bacteria we just mentioned, that if you have inflammatory acne, now the pores have gotten clogged, you have pimples, now that they're actually producing comedones, which are infected pores, infected follicles, and so we have these bugs in there that are living on the glycerol from the sebum, right, and causing and secreting their propionic acid, which is going to cause inflammation. What can we do about it? Treatment with vitamin A analogs, retinoids. Isotretinoin is a commonly used retinoid for that. Changes the pace of the keratinocyte life cycle, so it slows down turnover of those keratinocytes. Use antibiotics to try to knock back some of the bacteria that are present. Clindamycin, doxycycline, even erythromycin. We've heard about prescribed for that. Benzoyl peroxide is a commonly used agent that, number one, kills bacteria, and it also will help loosen up the material that's clogging the pores. So that's been a strategy. Scrubbing pads with benzoyl peroxide are a tremendous boon for people that have acne, at least moderately inflammatory acne. There's a new idea of using intense brightness, I guess you'd say, visible blue light wavelengths to kill the bacteria. The jury is still out on whether that actually does much, more than just using the isotretinoin and the antibiotics and the benzoyl peroxide. Some papers swear up and down it works 85% better, and some say that doesn't make any difference whatsoever. So I read a long review of it. It's just not clear whether it really works or not. But if a person, there's a lot of variability in members of the population how they respond to treatments. If it works for you, and you're someone who's tried this, I don't mean to say it's not possible, it's not true, or what you're saying isn't true. Very possible. Again, some studies have shown some incredible results from some people. Usually they're very small studies, but hard to generalize from. Nodular cystic acne is just very deep abscesses forming in the pores and all over the place, and a little bit more concentrated isotretinoin to try to um, get that to settle down a little bit more and use antibiotics and so forth. Picture of a familiar looking teen face. Whole different area. Now we're talking about a rash produced by Rocky Mountain bottom fever causing bacteria, Rickettsia, and it's transmitted by ticks. It's a small aerobic gram-negative bacteria. It can exist as a coccus, can exist as a rod, and even as like a thread-like structure. So it's kind of pleomorphic. Pleomorphic just means it has many different possible shapes. It's an obligate intracellular parasite, though. Can only live in the body inside of host cells. Produces a severe flu-like symptom and a non-itching macular rash, meaning remember the flat red spots, including on the palms and soles. They wanted to distinguish that between this and varicella, the virus that causes chickenpox, which does not produce any of its lesions on the plantar and palmar surfaces. So it goes into endothelial cells, and it can survive in those cells and grow. So it's basically killing the endothelial cells that line blood capillaries, and then they kind of leak out tiny microscopic amounts of blood and form microscopic but tiny amounts of blood and cause these regions of hemorrhage, and that's what causes the rash. It's actually little mini hemorrhages forming in the skin. Treatment. You need to use those antibiotics. If you go back through your antibiotic chapter, you'll remember we highlighted some that were able to penetrate into cells. A lot of antibiotics work outside of cells where the majority of bacterial infections occur, but these ones listed here are ones that can get intracellular and go in after these rickettsias. How about some viruses? Warts, papillomaviruses cause warts. And there's about, 45 or more strains. I may be behind on that already by now. 45 or more strains of papillomaviruses that have different properties. Some of them produce warts more on the hands. Some of them are more likely to produce genital warts. Some of them actually lead to, ultimately, cervical cancer. Ways you can get rid of warts. Cryotherapy, meaning freezing them. Use some liquid nitrogen and actually just kill the tissue by a very, very cold freezing. Liquid nitrogen boils at something like, I think it's minus 163 degrees Celsius. Pretty impressive. So get it nice and chilly. Salicylic acid can be used to degrade and etch away the tissue of the wart and reduce it down. Different ways of treating it. Imiquimod stimulates gamma interferon production, which will help the immune system go after virally infected cells and try to keep them at bay. Bleomycin is a nasty anti-cancer chemotherapy drug, but it's been found that if you inject that directly into the wart doesn't have any circulation in it, right? It's part of the epidermis, and you can inject that right into the wart. It's not going anywhere. It's not going to poison the body, but it can kill the cells in that part of that growing hunk of tissue on the surface of the epidermis. Excuse me. Poxviruses. Smallpox, a terrible disease that we mentioned just because it's historically so terrible to remember back to, and smallpox has a very high mortality rate and killed just millions of people throughout the world for a long, long time until it was finally eradicated. The one disease which we've successfully eradicated by just using good medical technology, vaccines. Utterly eradicated. Great big victory in public health. Monkeypox is a thing you probably hear about nowadays. There's a monkeypox outbreak kind of growing in severity, or in incidence, I guess how you should say, or prevalence around the world. And so that's been in the news a lot. It's a related virus. So we've been talking about possibly there is a monkeypox vaccination that could be used, and we're talking about trying to ramp that up and get that out to people around the world to try to slow down that monkeypox epidemic. if you could call it that. There's a picture of smallpox lesions, which you'll never see, happily, because no one will ever get smallpox again unless the most incredibly, unbelievable, dire thing happens to this few remaining samples of smallpox in laboratories in this country and in Russia, left over from scientific work. Chickenpox. Chickenpox is caused by a virus that has a bunch of different names, but varicella is one name of it. Herpes zoster is another name of it. Varicella zoster, you can also call it. Human herpes virus 3, I think, is the best thing to call it. It's a much more organized way to talk about the different viruses. There's 8 human herpes viruses, and if you can learn them, it's very helpful because it's a lot easier than remembering several common names, I think, for each one. But you should know these because they're so popularly used by people throughout the industry, the healthcare industry. So anyway, varicella causes chickenpox, and it's a childhood disease with a it's a vesicular rash, a little blistery kind of a rash that eventually crusts over. One problem with it is that after the disease is resolved, then the virus can remain alive. Well, to whatever degree it's alive, it can remain. Virus particles can remain in the dorsal root ganglia of the nervous system, and then it can reactivate. There's a vaccine for varicella, for chickenpox, and most of you probably never had chickenpox because of the vaccine. Occasionally, people will break through, quote unquote, people that have been vaccinated. 0.04% of the population that's received 2 varicella vaccines can still get the disease. So the vaccine is 100%. It's only 99.6% effective. Is that nice? I'm saying that right? 99.96, I guess, percent effective. Pretty good, huh? There's a little picture of a sensory neuron in the skin, and there's its long axon on its way. It's kind of a strange way to draw because got his body over here, and we were talking about his spinal cord over here. But anyway, the spinal cord, the peripheral nerves attached to the spinal cord by dorsal root and ventral root, and the dorsal root right here is where the cell bodies of the sensory neurons lie. It's called the dorsal root ganglia. And so that's where those human herpes viruses tend to reside when they go into latency, and they can be latent for 40, 50, 60 years. And then they can become reactivated. So there's a picture of a chickenpox rash. Shingles is what happens if that same varicella virus, or herpes zoster virus, as it's usually called when you're talking about shingles, is reactivated, and it will come back down the axon of that neuron to the surface of the skin, and it'll cause a lesion in the skin. Acyclovir is an antiviral agent. It's a nucleoside analog that will end up screwing up DNA replication for the virus, or synthesis to make virus particles. So, very important drug for several viral infections. I have to mention, I misspoke. I think it was last chapter or the chapter before. I read an article that suggested that acyclovir is a substrate for an enzyme that phosphorylates it, a viral enzyme specifically that phosphorylates it and produces the, I think it's guanine, it's an analog of guanine nucleotide. So you start with acyclovir that's not phosphorylated, add a phosphate group, now it looks like a guanine nucleotide, and it gets picked up by not the cellular DNA polymerase, only the viral DNA polymerase picks it up, and then the DNA polymerase, once it adds that acyclovir analog, it can't elongate the chain anymore, and it stops DNA synthesis. So it's a competitive inhibitor of guanine nucleotides, but it blocks the viral DNA polymerase. And I had said both viral and cellular DNA polymerase. Websites now this time, and I'm pretty sure the consensus is no. It's the virus. These human herpes viruses do not use the host DNA polymerase at all. They have their own, and that's what makes their copies of their genome. So here's a little diagram showing the reactivation of the virus. Now the virus has been dormant or latent in the dorsal root ganglion, the cell body of some neurons there. Now it's going to activate, and a whole bunch of new virus particles are being made, and they're all going to come down the axon. There's actually a little transport pathway right down the axon of the neuron to the surface and cause a skin lesion. And this is the pattern of where the sensory neurons from this particular dorsal root ganglion, all the sensory neurons that come to this level of the spinal cord, that's where they provide sensation to the skin of the body, and that's where all the viruses come out onto the surface. So it produces these patterns that are reminiscent of where the sensory neurons actually lie. They're dendrites. Very painful disease. The problem isn't the pain. People can bite a piece of wood or whatever it is and get through the pain. The problem becomes that the pain doesn't subside after the resolution of the viral lesions. You wind up with this neuralgia, this long-lasting pain sensation that can last for years, make people very, very uncomfortable. So it's a problem. So there's new vaccines for shingles. So people that have, even though you've had the chickenpox vaccine, as you get older, you might want to think about getting a shingles vaccine just to make sure you won't be able to get this awful sequela to chickenpox. All right, so we'll close it down, and we'll come back in just a little bit and look at the second half of chapter 21, diseases of the